Welcome to Biot Anime. Protein folding. Protein folding is the physical process by which a linear polypeptide folds into its characteristic and functional three-dimensional structure. Each protein exists as an unfolded polypeptide or random coil when translated from a sequence of mRNA to a linear chain of amino acids. This polypeptide lacks any stable three-dimensional structure. Amino acids interact with each other to produce a well-defined three-dimensional structure, known as the native state. Failure to fold into native structure generally produces inactive proteins, but in some instances misfolded proteins have modified or toxic functionality, example prions. Driving force behind protein folding. 1. The hydrophobic effect. Minimizing the number of hydrophobic side chains exposed to water is an important driving force behind the folding process. 2. Intramolecular hydrogen bonds. 3. Ionic interactions. 4. Disulfide bridges between neighboring cysteine residues. Anfinson's dogma. Anfinson's experiments indicate that the formation of higher-order structures is mainly dependent on the nature and type of amino acids present in the protein's primary structure. For his experiments, pancreatic ribonuclease enzyme RNase A is taken. RNase is an extracellular enzyme that possesses a single subunit that contains 8 cysteine residues involved in the formation of 4 disulfide bridges. The tertiary structure of RNase is stabilized by the formation of both covalent and non-covalent interactions. RNase enzyme is then subjected to denaturation using beta-mercaptoethanol and 8.0 molar urea as denaturing agents. The reducing agent was added in order to break the 4 disulfide bonds that stabilize the structure of ribonuclease A. Urea causes proteins to unfold. At high concentrations, urea alters the extensive hydrogen bonding network between water molecules, since urea contains both hydrogen bond donors and acceptors and thus interferes with the forces that promote protein folding. In experiment 1, Anfinson took the samples and used dialysis to remove the denaturation agent. The protein returned to its native structure. Experiment 2. He removed beta-mercaptoethanol first and sometimes later urea. Observed that an inactive native protein structure is formed. In the third experiment, he again added a tiny amount of beta-mercaptoethanol to the above solution and removed after a while, which makes the protein to return back to its active native structure again. Thank you for watching the video. For more videos, please subscribe to this channel and press bell icon. See you again in the next video.